Bismillahirrahmanirrahim. Now time to revise ketone body synthesis, which is very high yielding in case of starvation or in case of diabetic person. So when there is glucose level is low, acetyl coenzyme are not utilized by TCS cycles because oxaloacetate, which is intermediate, which is very important regarding TCS cycle, it start to produce gluconeogenesis to produce glucose. In this way, acetyl coenzyme start another cycle, which is ketone body synthesis. Because carbohydrates are necessary for oxaloacetate, but in case of starvation or diabetes, so oxaloacetate are less in numbers. So in this way, ketone body synthesis started. Acetyl CoA with the help of oxaloacetate normally start TCA cycle and form the citrate. But when there is low glucose, it start gluconeogenesis and TCA cycle stop and other cycle started, that is ketone bodies formation. Very important information regarding glucose has ability to store in form of fatty acid, but fatty acid is not ability to produce glucose. It has ability to generate acetyl CoA, but it's not have ability to produce glucose. So in this cycle, you can say that two acetyl CoA combine to form acetoacetyl CoA, which is 4 carbon molecule, and with the other acetyl CoA, it generate HMG CoA, 3 hydroxy 3 methyl glutaryl CoA, and now it's time to split the acetyl CoA, split down, and the acetoacetate formation, which is a ketone bodies, it is a beta ketone, so it is very unstable and it spontaneously break down into acetone with the decarboxylation. CO2 is removed, so 3 carbons are remain, and other if NADH is available, so NADH provide them hydrogen to attach the hydrogen at this level. In this way, the 3-hydroxybutyrate is formed. In this cycle, we have generated mainly three ketone bodies: acetoacetate, 3-hydroxybutyrate, or acetone. Acetone is we can smell them in breath because it has a fruity odor. So when some person is in condition of starvation, the fruity odor may be in their breath. So while the acetoacetate utilized to 2-acetyl CoA production and which finally produce 23 ATP, while on the other hand beta hydroxybutyrate generate 3 ATP. In liver, acetoacetate and 3-hydroxybutyrate are produced, but they are not utilized by liver. They are utilized by extrahepatic tissue, which may be peripheral tissue like muscle or brain cells or other tissue. In the 3-hydroxybutyrate, with the help of giving them hydrogen, so provide NADH finally converted into acetyl, which is reversible. Which is reciprocal to our last step. The acetoacetate with the help of thiophorase enzyme converted into acetoacetyl CoA, but it react with succinyl CoA, acetoacetate, to produce this acetoacetyl CoA, and the acetoacetyl CoA react with CoA to produce 2-acetyl CoA. In this way, 2-acetyl CoA are produced, and the enzyme which regulate this is thiolase enzyme. The very important concept regarding this slide is the thiophorase, which is present only in extrahepatic tissue. So in peripheral tissue it is absent in liver, so liver is unable to use ketone bodies. Uh, Ketone bodies are fuel for extrahepatic tissue, brain and muscle, and the acetoacetate first activated by transforming CoA from succinyl CoA. Then it's split into 2-acetyl CoA by the thiolase reaction. The very important concept regarding the pathological condition is ketosis. Mean there is ketone bodies level increases in blood, so which lead to ketonemia. Mean ketone body is high in blood, that will lead to acidosis. When there is ketone body is high in blood, so this blood when supplied to kidney, so in this case ketone bodies also increase urine, and that is ketonuria, that lead to dehydration. Why dehydration? Because it is osmotic, because we are losing salts or ketone bodies, that will also lose water, that lead to dehydration. It is very important to drink water when in condition of starvation to compensate this dehydration and acidosis. It lead to low pH. Means it is very important, it is very pathological condition, important pathological condition in a diabetes person. So it conclude our revision regarding ketone bodies as a fuel source.